Hello and welcome to all my dear students. I hope you are doing fine and must be having good time at home. So far beta, we have studied first two lectures of genetics part 1 in which we have discussed the details of Mendel's monohybrid cross and the laws framed on that basis, that is law of segregation and law of dominance. Apart from this, you also saw that Mendel's law of segregation is a complete law without any exception. It is 100% accepted. But if we talk about the law of dominance, then each postulate of law of dominance has some or the other exceptions. And in that category, we studied 4 exceptions: pleiotropy, incomplete dominance, codominance, and multiple allelism. I hope all those things which I have told you in my past 2 lectures are very well understood by you all. If you have not gone through the first 2 lectures, make sure that first you go through those lectures and then you watch the 3rd lecture so that sequence is maintained and you understand all very simple concepts also very well. So let's start with today's class. In today's class, we are going to study about Mendel's another work under the name Dihybrid Cross. So let's start with Mendel's Dihybrid Cross. Beta, like in monohybrid cross, we have seen that in monohybrid cross, Mendel studied inheritance pattern of one gene controlling one character at a time. Now, in dihybrid cross, Mendel tried to study inheritance of 2 genes at a same time controlling 2 different characters. The characters he chose, among the 7 characters which he initially chose. So from those 7 characters only, he obtained number of dihybrid crosses. One dihybrid cross here I am going to show you, and with this you can understand that how other dihybrid crosses would have also been done by Mendel. Di means 2. That means here we are going to study the pattern of inheritance of 2 genes simultaneously from the parents to offspring. For example, I am considering 2 genes controlling 2 different characters for my dihybrid cross as the shape of the seed, that is round and wrinkled, and color of the flower, that is yellow and violet and white. So cross involving 2 genes controlling 2 characters is a dihybrid cross. So basically I am going to consider the 2 genes, that is R gene and Y gene. Y gene signifies the flower color for pea plant and R signifies the seed shape. Now these 2 characters beta will have two contrasting traits. These are the 2 characters. You know, each character is represented by 2 contrasting traits. One dominant pure line, one recessive pure line. So for seed shape, the dominant pure line is this, which is having round shape of the seed, and this is the recessive pure line for seed shape having wrinkled arrangement. If I talk about flower color, then this is the dominant homozygous pure line. The flower color will appear violet, and here the flower color will appear white. So these are the pure line. See, these are the genotypes independently which I have written, and these are the phenotypes that I have mentioned independently. Now, these 2 characters which I have mentioned separately, but Mendel studied the inheritance of both the genes located on the chromosome together to check that what is the inheritance of 2 genes when inherited together. Now, beta. Okay, so first to start the cross, the first step is to select the parents, and parents always has to be pure line. So first step is selection of pure line parents. of which one has to have homozygous dominant pure line and another has to have homozygous recessive pure line. Correct? Chalo. So we are going to select our pure line parent. Now, suppose this is my male plant, which is homozygous dominant for both the genes. That is, this is the genotype. So phenotype of this plant will have round seeds with violet flowers. Correct? Now, this male plant will mate to do cross-pollination with homozygous recessive pure line for both the genes. So the genotype will be this small r, small y, small phi, and phenotype will be wrinkled seeds with white flowers. Correct? Yes or no? So this is the genotype and phenotype of my parents. Now I have to perform cross-pollination. Cross-pollination means first obtain the gametes from both the pure line parents. So the next step is doing cross-pollination. And for cross-pollination, we have to obtain the gametes. Now if you check the genotype beta, then both the gene in both the parents are present in homozygous state. That means the number of gametes formed by both the parent will be only 1. As over here, n value is equivalent to 0. So 2 की power 0 means only 1 gamete will be produced by both the parent. You can see the genotype of male is also homozygous and genotype of female is also homozygous. No heterozygous gene present. So the value of n will become 0. Anything raised to power 0 is 1. Now let's check out the gametes from male and female parent. Now for male parent, if I want to prepare the gametes, what is the arrangement of gene? Yes or no? This is the arrangement of gene. So every time you are going to only obtain one type of gamete which is having dominant alleles for both the gene. So this is your gamete from male parent. Similarly, if I want to check the gametes from female parent, then what is going to be the pattern? See, this is the arrangement of gene. Yes or no? Both the genes are present in their homozygous state. So every time we are going to obtain only one type of gamete where both the alleles of the gene are present in homozygous recessive state. So this is the gamete. Which gamete? Female gamete. Now gametes you have obtained. Next step is fertilization. After cross-pollination, that means after crossing the gametes, then fertilization has to be done. And after fertilization, we are going to obtain our F1 generation offsprings, the first generation offspring from the parents. So what will be the genotype? You know, just make the cross. Right? All offsprings will appear. All offsprings will have round seeds and purple or violet color flower. None of the offspring, suppose even here also 100 offsprings are obtained in F1 generation after the cross between the parent, then all 100 parents will produce seeds in their pea plant, round, no wrinkled seeds. All flower will be violet, no white. You can see the reason behind. So this is the phenotype, as in case of genotype, both the genes are present in their heterozygous dominant state. Understood till here? Correct. Now, what is the next step? Same as we performed for monohybrid cross, the next step is to perform selfing. So after obtaining all the offsprings, Mendel wanted to check that where is the genotype of, or where is the phenotype of the female parent. All offsprings are coming round with violet flower. Why none of the offspring are bringing wrinkled seeds with white color flowers? Where is the female phenotype? Has female not done anything? So just to ensure that, beta, next step is he performed selfing to obtain next generation offsprings, that is F2 generation offsprings. So before that, we need to obtain gametes for F2 generation. Now, what is the condition? Both the genes are present in heterozygous state. So, the value of n over here will become 2, and root 2 ki power 2 means 4 types of gamete will be obtained. And I think you can calculate the types of gametes. Just do the branching method, beta. Right? So one gamete is having both the dominant alleles. Another gamete is where one dominant allele, another recessive allele. Third, one recessive, one dominant. And fourth, both recessive alleles. So these are the 4 types of gametes which we obtained after crossing. Now, one thing, beta, I would like to tell you. Just pay attention. Beta, both the genes are present on homologous chromosome. What am I saying? Both the genes controlling 2 different characters, they are present on homologous chromosome in heterozygous state. Like this. Correct? Okay. Now, what will happen during meiosis, during gamete formation? Meiosis takes place where crossing over is occurring. Okay. Now, crossing over means exchange of genetic material between the homologous chromosome. Now, out of the beta 4 gametes, this and this are the parental gametes, RY and small r, small y. These are called as parental gametes. Why parental gametes, beta? Can anybody tell me? Because they are same as the parent which were taken initially. Just check the parental gametes. Right? So these are the parental gametes. However, the other 2 gametes which we obtained are due to crossing over, are due to exchange of genetic material. So these 2 are called as recombinant gametes. Right? So these 2 are recombinant and the first and the last are parental. I have shown you the initially also the parents, they formed gametes like that, and the remaining 2 are due to crossover. So they are called as recombinant gametes. So we are obtaining recombinant and parental gametes in the ratio 1:1 for time being. Now what will happen? The same type of gametes will be produced by this parent also. Now crossing will take place. All these gametes will mate to fuse each other to form the zygote. Now, how many gametes are there? 4. Correct? So, how many zygotic, how many types of offsprings you can expect? 4 ki power 2. That means 16 combinations of zygote are possible. Right? Now, gamete square. To calculate zygotic combinations, simple formula is number of gametes ki power square. So types of gametes are 4. 4 ki power 2 means 16 types of zygote we can obtain. And to make sure that we are obtaining them, we have to draw a Punnett square. So yes, here I'll be I think this is going to be a small space. For Punnett square, we need a complete full 1 page. Okay, I'll just add one extra page. Yes, so here I am going to draw the complete Punnett square with 16 zygotic combinations. So I'll make a chart of 16 blocks. 4 horizontal, 4 diagonal. Correct? How I came to know that 16 zygotic combinations will be formed? Because I know the formula gamete ki power square. If 4 types of gametes are produced, then 16 types of offsprings can be formed. Now, what are the types of gametes? Just write it over here. Correct? Of which first and last are parental types of gametes. These are parental, and these two are recombinant types of gametes. Now, same gametes are produced by the other parent, which is female parent also. So, gametes from the female parent, you can write it over here. Right? Now, the gamete arrangement is done. You have to make the cross and obtain the what? And you have to obtain their zygotic combinations. So, just try to fill them. I am filling in my book or in my page. You can do it for your own self also. So just take some time. Till then, you can wait and you can revise it. Okay. Yes. R, capital Y, small y. Here, capital R, capital R. Okay, this done. Then completed. Okay. Then yes, here. Okay. Yes. Now, these are the different 16 zygotic combinations possible from my dihybrid cross, where I have chosen these two genes located on homologous chromosome. Now, beta, if you check these 16 zygotic combinations, okay, you know, initially Mendel was only able to see the phenotype. He was only able to observe the morphology. And then with the help of test cross, he calculated the genotypic ratio also. So after doing all this F2 generation zygotic arrangement, the phenotypic ratio for F2 generation which Mendel obtained was, see, 9:3:3:1, where 9 offsprings were having, I'll prove it with the help of box also, but first I'll be writing round and violet flowers. 3 were having wrinkled plus violet flowers. 3 combinations were having round and white flowers. And 1 combination was such where all were wrinkled with white color flowers. Clear? So this was the phenotype that he saw. Now let's check the phenotype. That how is this possible? So 9 you have to see round and violet. In this beta, in this round and violet, you are going to check both the types of genotype, where either R is homozygous dominant or heterozygous dominant, Y is either homozygous or heterozygous. So for this 9, for 9 phenotypes, that is round and violet, we are going to consider both the types of genotype where both the genes are either present in dominant state, homozygous or heterozygous. For remaining 3 beta, the genotype which we are going to consider is homozygous recessive R, whereas dominant Y, may be dominant homozygous or heterozygous. For next 3, we are going to consider R in heterozygous state genotype and Y in homozygous recessive genotype. Correct? And 1 is recessive for both the genes. So if you want to check these 9, you have to check such genotype where either both the R and Y are present in dominant homozygous state or even in heterozygous state. So chalo first calculate this 9 number. Okay, I'll just use some other color pen. So first, round and violet. Correct? Yes or no? See, both the genes are present in homozygous dominant. Correct. Second, so this is A. I'll just, yeah, wait. So I'll just name it as A1. This is for A. Here we'll count B, here we'll count D. Correct? So this is A1. This is also A2. Why? Because R is in heterozygous dominant, but Y is in homozygous dominant. So considered in my first genotype. Correct? Round and yellow, round and violet. Here also A3. You can see round and violet. Fourth also. How? Round and violet. Both are heterozygous coming in my first genotype. Fifth. This is fourth. Fifth. Round and violet. Correct? But if you see this combination beta, this genotype is matching my second, is fulfilling my second condition where R is in recessive state and Y is in its dominant state, maybe homozygous dominant or heterozygous dominant. So this is B1. This is B1. This A6. Next, fulfilling my second condition. R heterozygous, R is in homozygous recessive, Y is in dominant state. Maybe homozygous dominant, maybe homozygous recessive. So this is B2. Now, again, fulfilling my first condition. Seventh, we have to total make this 9. Still 2 are left. Next, 8th. Now, next one is fulfilling my third condition where Y is in its recessive state and R is present in its dominant state, maybe homozygous or heterozygous. So this is C1. Again, same. C2. This one is fulfilling my 9th. So now, first condition is completely fulfilled. Done. 9 total genotypes you have such seen, which is going to bring the phenotype round and violet. Now, this is fulfilling my second condition. So that means B3. Why B3? 1, 2, 3. So 3 condition fulfilled. Next, this is fulfilling my C3. Now you can see C1, 2, 3. Third condition fulfilled. And last is D1 where both the genes are present in their homozygous recessive state, fulfilling my last condition. Now can you see that the phenotypic ratio 9331 is absolutely correct, and we have proved with the help of the chart also. So this is the phenotypic ratio, and here I have written genotypic ratio in a minimalistic form. But now I am going to write the genotypic ratio in expanded form for dihybrid cross. Now, F2 generation genotypic ratio is beta this 1:2:1:2:2:4:1:1. Now this you have to decode by your own self. I'll just tell you one of the genotype for 4 is this, where both the genes are present in their heterozygous state. For 1, this is where both the genes are present in homozygous dominant state, where this is for both the genes present in their homozygous recessive state. Remaining 2, 1, 2 and everything you have to do it by your own. It is very simple beta. You have this Punnett square in your hand. You can use this Punnett square and surely you will get all the genotypes clearly check the Punnett square, write the genotype for each number, for each value which I have mentioned in the genotypic ratio. Will you do this thing? Yes or no? Now, this was all about the Mendel's dihybrid cross. So this is how you obtain the phenotypic and genotypic ratio. Of course, for phenotype, he has to do nothing. He could simply visualize. But yes, for genotypic ratio, he had to make the test cross for each offspring to calculate their genotype and to calculate this ratio. Got it? Yes or no? Okay. Now, one more thing I would like to tell you, beta. The phenotypic ratio for F2 generation is 9, 3, 3, 1. Correct? Where you see, beta, 9 are round and violet. Correct? 3 are wrinkled and violet. 3 are such with phenotype round and white flowers, and one offspring out of 16 will be such having wrinkled seeds with white color flowers. Correct? Now, if this is the ratio in which number of offsprings are obtained, then can't we say that the percentage of parental is to recombinant phenotypic ratio is 10:6? Why? Because this 9 number, round and violet, see, how were the initial parents which I took? How was the initial, where it is? Okay, so initially my parents were of phenotype round and violet, then wrinkled with white condition. These are the phenotype of my parent. Correct? So the ratio which you calculated at last, beta, yes, here. Can you see this 9? So this phenotype is similar to my parent. Similar to male parent, right? And the last one, this is similar to your female parent. That means out of 16 zygotic combination, 10 phenotypic arrangement will be similar to parent, and remaining 3:3, which you see in the center, is actually the recombinant phenotype, which arise due to recombinant gametes, which I told you in the beginning. So these are the recombinant phenotype. So you can say one thing, that the recombinant and parental phenotype are in the ratio 10:6. Clear? So of course you know parental type are more than recombinant type. So that means formation of parental gamete is surely more than recombinant gamete. So initially I said one thing, that all are produced in same ratio. Actually, I was not correct. Actually, the number of parental gametes is more than recombinant gametes. That is why at the end you see more number of offsprings with parental type and less with recombinant type. Clear? Understood? Will you be able to solve questions? Yes, ma'am. So this is all about the dihybrid cross of Mendel. Now, like in monohybrid cross, beta Mendel, after completing the cross, he proposed some laws on the basis of his observation. Similarly, after completing the dihybrid cross also, Mendel proposed a law called law of independent assortment on the basis of his observation. Chalo. So let's talk about his observation and let's talk about the law framed on the basis of Mendel's dihybrid cross, that is law of independent assortment. Now, first of all, let me tell you, this law is also not accepted. It is also having an exception by the name linkage. So, exception to the law of independent assortment is linkage. Will not tell about linkage right now, but yes, this linkage, recombination, everything is there in your syllabus, which we will do later. Right now, I have just introduced the term linkage, which is an exception to Mendel's law of independent assortment. Now, why so? What is this law? Let's try to understand. Now, beta, Mendel said that whenever two genes located on homologous chromosome are inherited together from parent to offspring. Again, I am repeating, listen to me carefully. It is said that when two genes located on homologous chromosome are inherited together, so during the time of gamete formation, during the time of meiosis, both the genes are going to segregate completely independent of each other. Correct? First of all, there will be separation of gene, and then in the next step, their alleles are also going to separate without any blending. Okay. This portion is okay. This is law of segregation, that when a gene segregate, it's both the alleles separate independently. Absolutely correct. But the first statement of the law says that both the genes, that means when more than one gene is inherited together, located on homologous chromosome, beta, they do not segregate independently. Mendel was wrong over here. When two genes, they are located on a homologous chromosome, they can influence each other. Correct? They do not segregate independently. R is going to influence Y. Y is going to influence R. This is linkage. Correct? This is called as linkage. That means if two genes are present on the same homologous chromosome, they are linked, they are associated, and during the time of gamete formation, they do not separate out properly. They do not separate out without blending. They are going to influence. Okay, later their alleles will segregate out, correct. But genes located on homologous chromosome do not segregate out independently during the time of gamete formation, and this is known as linkage. Details I will teach you when we will come to linkage portion. For time being, just try to understand that what is law of independent assortment. So what is written on the screen? According to this law, when two genes are inherited together, located on homologous chromosome from parents to offspring, then according to Mendel, both the gene segregate out independently, and later their alleles also segregate. Allele segregation is okay, but gene segregation is not okay. They are going to influence each other, and this is only known as linkage. Right? So for time being, I'll keep it till here, simple and short. Details of linkage will be taught you as an independent topic, but later on. So till here is Mendelism. Till here, monohybrid, dihybrid cross, you have seen of Mendel. Now you have seen the work of Mendel, the laws proposed by Mendel, and the errors made by Mendel. Correct? So till here is classical genetics, the genetics given by Mendel. Now we are heading towards post-Mendelism era. That means we are coming from classical to modern genetics. Like this Mendel is known as father of classical genetics. So post-Mendel, another scientist came by the name Bateson, who is also known as father of modern genetics. He added some more important points to enhance our understanding for genetics. Correct? So now we are going to study the post-Mendelism genetics, which was proposed by Bateson, also known as Batesonian genetics, right? Modern genetics. So he's known as Bateson, is known as father of modern genetics. This two gene interaction now we are going to see, which is a post-Mendelism effect. Two gene interaction. Now what Mendel said, beta? Mendel was always in the favor that one gene at a time can control one character only. Correct? Like R gene is responsible only for controlling seed shape. Y gene is only responsible for controlling flower color in pea plant. So Mendel was always in the favor one gene, one character. But you have seen the exception, pleiotropy, where many characters were controlled by one gene. Correct? Under the same heading of pleiotropy, Bateson proposed two gene interaction, where you will see two genes either located on same chromosome, means homologous chromosome, or located on different chromosome are controlling one character. Right? So what is two gene interaction? According to Bateson, beta, two genes either on same or different chromosome controlling one character. Like for example in Lathyrus odoratus. Lathyrus odoratus, beta, it is the scientific name of sweet pea. Sweet pea is also known as the scientific name of sweet pea is Lathyrus odoratus. Mendel worked on normal pea, garden pea, Pisum sativum. In the same family, there is one more pea called as sweet pea, Lathyrus odoratus. So on Lathyrus odoratus, the scientist Bateson worked and he saw that the flower color of Lathyrus odoratus is under the control, is under the influence of two genes, that is C and P. Clear? So what are the two genes which are influencing one character in Lathyrus odoratus? That is flower color. And what are those two genes? Those two genes are C and P. C and P genes are controlling one character which is flower color in it. Now see how. It is said that beta, flower color of sweet pea can be of two. There are two colors, white and purple. Purple color comes when both the genes are present in dominant state, either homozygous or heterozygous. Even if one gene is present in its recessive state, then flower color will change from purple to white. That means to bring purple color, in the flower, I'll tell you the reason also. Both the genes should be beta present in dominant state. Both the genes should be present in dominant state to bring the flower color as purple. Now, even if one of the gene becomes recessive, then the flower color changes to white. Why? Because to bring purple color, a pigment is required called anthocyanin, and production of this pigment is dependent upon the availability of protein synthesized by both the genes. Now what happens beta? This is C. There is a precursor to synthesize anthocyanin, which first need to change into precursor, and then precursor change into anthocyanin pigment. Correct? Now, to change first pre-precursor into precursor, dominant C is required, maybe in homozygous or heterozygous. Now, pre-precursor is formed, but to form anthocyanin, the precursor require dominant P gene. That means when both the gene will be present in dominant state, then only pre-precursor will change into precursor and precursor will change into anthocyanin, and then only the flower color will become purple or violet. Now, if beta, any of the one gene is present in recessive state, suppose C gene is present in recessive state, what will happen? Pre-precursor will not be able to form precursor. If precursor is not there, then what the dominant P will do? Because dominant P cannot affect pre-precursor. First precursor need to be formed, then only P gene can act to produce anthocyanin. Now, if suppose C is in dominant but P is in recessive, so precursor will be formed from pre-precursor. But who will convert precursor into anthocyanin? Again, no anthocyanin. So, color will remain white. But when both the genes are present in dominant, pre-precursor will first convert into precursor, and with the help of P-dominant gene, precursor will convert into purple color, anthocyanin. So, here you see one flower color controlled by two genes. Absolutely understood? Correct? Now, Bateson beta, he explained the this effect, two gene interaction in flower color of Lathyrus odoratus under the name complementary gene interaction. So, this two gene interaction, which you see in case of flower color of Lathyrus odoratus, is known as complementary gene interaction. Clear? Yes. What is complementary gene interaction? Cool. Very good. Now let's try to make the cross to understand it in a better way. Now beta, cross is absolutely made in the same manner as Mendel used to do. First step is selection of parent. Parent has to be pure line. So these are my parents. So purple color, homozygous dominant crossed with homozygous recessive. Gamete over here will be capital C, capital P. Here gamete will be small c, small p. Now, combination. All flower will be purple. So, till here, everything was normal. As Mendel said that all offsprings in F1 generation will bring the phenotype of only one parent out of the two. So, this is actually F1 generation. Till here, everything was right. But then, when he tried to make the selfing, जब इन्होंने selfing करवाई, then this person, this Bateson was expecting the ratio 9331. Why? Because it is a kind of dihybrid cross. You can see two genes are involved. So as Mendel said, when two genes are inherited together, then the ratio for F2 generation phenotypic ratio is going to be 9331. Bateson thought that okay, 50% of the experiment is okay, following Mendel law, then remaining 50 should also be okay. And after performing selfing, after obtaining F2 generation, phenotypic ratio should be 9331. But the ratio was not 9331. Let's see what the ratio will be. Now, what will happen? You are going to obtain the gametes. Gametes will be 4 types. So, total 16 zygotic combinations. Yes or no? 2 heterozygous gene. So, how many zygote? Gamete 4. 2 की power 2. 4. Zygote 16. 4 की power 2. So, these are the gametes. Right? These are the gametes. Will you fill this Punnett square for me? Yes, I leave this Punnett square on you. You have to fill this Punnett square. I'll just tell you the results directly, which is very, very important now. Beta, this Bateson expected ratio for complementary gene interaction as 9:3:3:1. That means what he was expecting out of 16 zygotic combinations, 9 will be like this, having purple color flower, then remaining 3 will be, and then here it will be this, right? He was expecting the ratio in this manner, but the ratio changed. Why? Because these 9 will be purple. However, all these remaining 7 types of zygotic combination will produce white color flower. How? Because you see, in case of this second and last, both the genes are not dominant, and you know, if both the genes are not dominant, flower color will not come purple, or rather it will remain white. So instead of getting 9:3:3:1 ratio, the ratio will change into 9:7, where 9 will be purple type and 7 combinations will be white type. Clear to everyone? Why 7 white? Because you see, at least in this, C is recessive here, P is recessive here, both are recessive. So only first case purple color flower where both the genes are present in their dominant state. Understood? Yes or no? The complementary gene interaction. If yes, then Bateson also gave one more example to explain two gene interaction under the heading duplicate gene interaction. Here, beta, in duplicate gene interaction, you will see that the two genes A and B are controlling the capsule, the fruit shape of shepherd's purse, which is Capsella bursa pastoris. So fruit means capsule shape. Shepherd's purse is basically Capsella bursa pastoris. This is a scientific name for this. Is a common name, shepherd's purse, and Capsella bursa pastoris is the scientific name. Correct? In this, the capsule shape, the fruit shape was studied by Bateson under the heading duplicate gene interaction in two genes. Why? Because there are two genes, A and B, controlling one character, which is capsule shape. Now, if you check this beta shepherd's purse, then in shepherd's purse, there are two shapes of capsule. Some are triangular shape and some are top shape. I will not draw. Yes. So some are triangular shaped, triangular. And some are top shaped capsule. So the shape is controlled by two genes. Now, what is the explanation for duplicate gene interaction? Duplicate gene interaction says beta to bring triangular shape both the genes, or even if one gene is present in dominant state, then triangular top shape will come. However, if both the genes are present in recessive state, then capsule shape will become top. So that means to bring triangular shape in the capsule of shepherd's purse, this genotype or this genotype or this genotype, all the three are going to bring triangular shape. Either one of the gene or both the genes, if they are present in their dominant state, the shape of the capsule will become triangular. The only condition to bring top shape is when both the genes are present in their recessive state. Then only the shape of the capsule will come top. So, in this case, the ratio will get modified how? Here beta, the ratio 9:3:3:1 will modify as, see, this is only we write now. This Mendel taught us. So we are going to follow Mendel over here. So you see the first 15 types of zygotic combination are having either both the gene dominant or even one gene dominant. So they are going to produce beta triangular shaped capsule. But last, when you see this, the last zygotic combination, the 16th zygotic combination is only the one to produce top shaped capsule where both the genes are present in their recessive state. So for duplicate gene interaction, ratio for the dihybrid cross will change from 9:3:1 to 15:1. In complementary gene, you saw the ratio as 9:7. Here, the ratio is going to become 15:1. Understood? These are the two simple examples to show that if two genes inherited together from parent to offspring controlling one character, then the ratio is not of the Mendel's dihybrid. However, it gets changed accordingly. In complementary, the ratio become 9:7. In duplicate, the ratio become 15:1. Right? Yes or no? Okay. Now beta, apart from this two gene interaction which you have seen in complementary and duplicate, there is one more type of two gene interaction under the name epistasis. Today, I am just going to introduce this term epistasis and the types of I'll just explain the term today. The crosses on epistasis basically that we are going to do in our next class. So today I'm just going to introduce what is epistasis and how it is different from Mendel's dominance. Correct? See, epistasis means when two genes either on same or different chromosome are influencing one character, then one gene can suppress the effect of another gene even if the other gene is present in its dominant or recessive state. Getting my point? What Mendel said, beta? Mendel said that a gene located on chromosome represented by a pair of allele. According to Mendel, one gene which is represented by two alleles. Now, if both the alleles are present in heterozygous state, then one has to be dominant, correct? And another has to be recessive. One has to be dominant, one has to be recessive. The dominant allele is going to express and will suppress the recessive allele, correct? So basically Mendel dominance was intragenic. Mendel's dominance is beta intragenic. That means within one gene. See, this is only our gene. This is our gene. And within our gene, you are seeing one is dominant, another is recessive. You can also say it as interallelic. In between two alleles, capital R, small r, two different alleles. So interallelic influence. But with respect to gene, only one gene. So within the same gene, intragenic is dominance. Now, Bateson introduced the same concept but in between two genes, that is epistasis. So, it is intergenic, also called as non-allelic interaction. Why? Because in case of beta epistasis, what we see, first of all, there are two genes influencing one character. There are two genes together influencing one character. Right? So here two genes R and Y, between two genes intergenic, correct? Now, maybe R is suppressing the Y, or maybe Y is suppressing the effect of R. So it is a kind of dominance only where one is suppressing the effect of another. But still it is different from dominance because dominance is with respect to one gene controlling one character. Epistasis is with respect to two genes controlling one character. Right? So what are you going to write? In case of epistasis, one gene is influencing, or you can say is suppressing the phenotypic effect of another gene. Right? Now, the gene who is suppressing, suppose R is suppressing the effect of Y. So, the gene which is suppressing is known as epistatic gene. Like here we use the term dominant and recessive allele. Dominant, the one which suppresses. Recessive, which is getting suppressed. Here, the one which suppresses is known as epistatic gene but not dominant gene. And the one whose effect is getting suppressed is not known as recessive. Rather, it is known as hypostatic gene. Clear? Understood till here? Now, beta, epistatic gene can be either present in dominant state or can be present either in recessive state. On that basis, we have two types of epistasis, dominant epistasis and recessive epistasis. So on the basis of the nature of epistatic gene, we have two types, dominant and recessive epistasis. Dominant, what is this? What is recessive? Today I am not going to discuss because we have done a lot in last 3 lectures. I want you all to revise and solve questions till here. In our next class, we will understand more about epistasis, types of epistasis with the help of example. Clear? Yes or no? So now you are having some homeworks, some questions for homework. Please do them. Make sure you are always doing the homework. Plus, you are solving the questions by your own also, beta. This is very, very important. Take these classes seriously. As I always say, these classes are there to support you, to help you in your NEET preparation. So please take them seriously as we are doing. Hope you understand me well. Thank you for joining me. Bye-bye, everyone. Take care.